There we go. Can you hear me okay?
Yes.
Great. So it's great to be with you virtually and to give a talk to start up your great sounding conference off today. So what I was going to do is just basically talk about my work over the past recent times and kind of a lot of background about how I got into that and where I want to go with it, I guess. And I'd like to have like questions, make it interactive if you want, just ask questions if you think like, how did you end up doing that or something like that? Just ask and we can get into that and go off on a tangent because I think tangents are great. So I've called it BioArt and Bacteria and the Collateral Effects of the Pandemic. So what I mean by collateral effects is these kind of hidden and unforeseen and unexpected things that have happened, some good, some bad, and I'm exploring that as well. So, and that's the recent work. So let's just click on. So these are some of my roles. I've been working in this field for about 20 years now. I think Danielle already asked in the chat if I could define BioArt, because I work in this kind of field of BioArt, sculpture. I trained in fine art. I didn't train in science as such. I did science a bit at school, but it wasn't taught in a way that was really engaging. So I didn't really enjoy learning about science in school, but I was always interested in some of these scientific ideas, but not really responding to the way it was taught in school. And so I went and did a fine art degree and a fine art master's degree, and also some research in the arts as well. And this led me to work in this way with this form of art called BioArt, which is a term that was coined originally by Eduardo Katz. It kind of means art made with or about living things. There are lots of different definitions. Some people are really like strict on it and say it has to be like actual living art. Some people say it has to contain living things in its process or somehow, or other people just use it to term things to do with biological art. So art to do with, you know, anatomy or nature or something like that can also be BioArt by some definition. So I'm not going to strictly define it because I think you can find your own way. I talked to Eduardo Katz and I asked him if he was happy with me using the term that he defined, and he said he was. So so that's where I'm sort of starting. The curator, who's an American curator called William Myers, kind of defined it in those quite loose terms that I talked about, and he wrote a very interesting book, if you want to see more of it, called BioArt Altered Realities, which is a lovely big glossy book on the subject. But anyway, I digress. So these are some of my many strange and interesting roles. I'm a freelance artist, so I'm a practicing artist, and I have a studio where I make my work, but a lot of my work is also made working in laboratories. So I have a role with modernising medical microbiology at the University of Oxford, who are scientists looking at whole genome sequencing of bacteria, and they've been doing this for many years, and actually their research into genomics of infectious diseases has really fed into the work that was done at Oxford during the COVID-19 pandemic. So it's kind of, some of them are very senior scientists there that I'm lucky enough to work with. And then also something, a place called the National Collection of Type Cultures. Now it sounds like something to do with culture, national collection. It's cultures of bacteria. It's the world's oldest collection of pathogenic bacteria. So bacteria that make you ill, basically. And they have a collection for that. And these are very historic microorganisms. So they have, you know, samples of plague. They have, well, I'll go into details of it, but they have all sorts of fascinating things. It's a kind of the ideal role for me. I also love computer science and working with robotics, and I'm an artist in residence and visiting research fellow at the University of Hertfordshire, also Brighton and Sussex Medical School. I was the 2018 president for the Science and the Arts section of the British Science Association. So I'm really passionate about this thing of bringing art and science together and kind of using that as a lens through which to explore and investigate the world and communicate issues. And lots of other things. So at Boku University in Vienna, working with, recently finished working with Schindler in Lucerne in Switzerland, working with an EU consortium about chicory plants and plant science and genetic modification of plants, because that's not something that's done in Europe. It's something that happens a lot in America and is considered normal there, but not so much in Europe. So we're interested in exploring what kind of levels people are comfortable with and things like that. So these are all some of my multifarious roles, and another important one at the moment, I'm going to go off and do a residency shortly, finally getting to travel a bit again, with the Helmholtz Institute of Epigenetics and Stem Cells in Munich shortly. So how did I get into all this? Well, I was kind of fascinated in bacteria, really, but the way I got into that, and this is going to be something, this is, you know, for you, for people listening here, you think this sounds like ancient history, but it's not so long ago. It's about 20 years ago, when you were probably kids or just about to be born or whatever. The World Wide Web became accessible to ordinary people like me. You didn't have to work in a university. You maybe had a computer at home for the first time, so you could start to search for things, but search engines weren't anything like they are now. And before that, you only kind of had access to information that came through the media. So like TV, advertising, TV programs, newspapers, things like that. That was where you got your information, unless you wanted to go off and actually go to a library and wade through books that you wouldn't necessarily wade through. You wouldn't stumble upon kind of information. You couldn't kind of follow those routes very easily. But around that time, it became possible in sort of the mid-90s to, and a bit after that, sort of the year 2000, around then, to kind of not Google's the wrong term because that wasn't around either, but look up things on the internet and search for things and find things out. And I started to realize that these E. coli bacteria that I was reading about in the newspapers that cause diseases were actually integral to our immune systems, into our health, and they helped us digest our food. I stumbled upon this research, and I thought, well, that's so weird. The difference between what the scientists get to know and what normal people get to know. And it was a completely different story. It was this story of this devil bacteria that we must get rid of versus the thing that we need to live. And I was thought, how come this story isn't kind of shared? And there was a TV show in the UK around that time. And I mean, most countries I've given talks at, they say that they've seen similar shows. So I think it's a sort of syndicated kind of program. It was called How Clean Is Your House? And people would go round, like these crazy cleaning ladies would go around with microbiologists and start kind of swabbing people's houses, and then go things like, there was an episode where they said, there's a species of Yersinia on this pillow of yours in your house, and that's a similar strain to the one that causes plague. So these people are completely confused at this point and thinking they've got, the Black Death growing on the pillows in their house because they're so dirty. I mean, that's not true. There's environmental strains of Yersinia and things. So they were like playing with that and exaggerating it. So I got fascinated with the idea that everything in my house was covered with bacteria. In fact, pretty much everything in the world, even sterile things, have some bacteria on it. It's really hard to totally sterilize something. So, and even if you have the bacteria already going on it again afterwards, different kinds of bacteria. So I was fascinated in that and started to kind of swab my home and try and culture the bacterium from it. And the way I managed to do this was by, I sent an email, also in the fairly early days of email, I sent an email to the medical school in Brighton and said, I'm an artist, this is the kind of work I do, and I'm really fascinated in microbiology, and I would love to speak to some scientists there, or scientists there who'd be willing to talk to me. And I got an email back from a scientist, and she said, well, I don't do bacteria, but I do allergy, and they're kind of related, but I really like your work, so maybe we could do a project together. So I didn't go, oh no, it's not bacteria. I was like, yeah, let's do a project. And we managed to get some funding and do an amazing project, actually, where we went and worked with a school, and we turned the whole school into an art installation for an evening. So we had art interventions around the entire entire school and the young people taking visitors around the school on tours to experience kind of the whole thing. And this is an image of an artwork that I made that was based on swabbing bacteria in my house, well, swabbing the chair and swabbing my bed. And the images are textile works based on those bacteria, because the scientist I was working with came to me one day and she said, "Anna, I've managed to find you a microbiologist to work with, but don't do that thing you do where you come and blurt out all your crazy ideas at him, because he's quite normal and you'll frighten him off." And so I went to meet the scientist, and it turned out he's not at all normal as she claimed him to be, and actually had his own arts practice on the side. And his wife, who unfortunately died a couple of years ago, said to me, he was just waiting, you know, to meet an artist and to have this kind of projects and stuff, because that was that side of him. So he was in the end became quite a leading microbiologist in the UK, and now he's retired, but he retired early. But he's writing up books on the natural history of bacteria and all sorts of fascinating things, and still doing research. So this was a kind of my answer to how clean is your house, which I consider to be more like how sublime is your ecosystem. You've probably all heard about this term, the microbiome, which means the sort of the bacteria that live on you and in you. In those days, it was called normal flora, and it was considered to be of no commercial or medical interest. So these environmental bacteria that live all around us, nobody cared about, because we didn't have the tools to study it. And it's all about the tools, the development of science, and what things are interesting to science and things. There's a lot to do with the tools. So this image here, I then was introduced to work with modernising medical microbiology, and we were working with MRSA, MRSA as we call it in England, methicillin-resistant Staphylococcus aureus bacteria, because there was a lot of this drug-resistant bacteria, and there was a big outbreak in the local hospital. So I started to study that and make art with it. And I was working with a researcher who was studying it, and he said to me one day, he said, and it grows this amazing denim blue when you put it on a petri dish. And this was kind of music to my ear. So we started making experiments. I was like, do you think we could grow it into cloth and kind of pattern that cloth with it? So these are kind of early experiments on this chromogenic color changing agar, which makes MRSA bacteria grow blue. And you can see these sort of white paper bits, these little dots and these strips here. These are antibiotic impregnated paper, vancomycin susceptibility disc, and an MRSA detection strip. And the bacteria is not supposed to grow up to them. And you can see this, what they call a zone of inhibition, meaning that it can be treated with vancomycin. So we used that to kind of pattern things. And and we had no idea that this would work, but we can't exhibit live superbugs. So we had no idea if it would work, but we decided to try and sterilize it in high pressure steam sterilization and see if it would keep its color and wouldn't fade. And it worked. We were pleased to see that it worked. And then we made this quilt out of the squares, and all the blue here is the bacteria growing on the this special color changing agar, and the polka dots made with vancomycin susceptibility discs. And the white square there is as if vancomycin was an intravenous drip into the body. And the yellow embroidery is turmeric, which is somewhat antimicrobial, but not enough to be good to treat MRSA, as you can see from it. And why am I interested in antibiotic resistance? Because it is a huge existential threat to humanity. As we've seen with like COVID, we've seen the impact of infectious diseases. And I've always said in my talks, the reason I'm so fascinated in infectious diseases is because of the huge impact that they've had on the world throughout history, and that they affect us all. We've all either experienced some sort of infectious disease, or we know someone who has. And we all have our stories to tell, and we're all being kind of affected by them all the time, in small ways, in big ways, but anyway, it's there. And I went on to study this subject further working with modernising medical microbiology, and they were studying at this point whole genome sequencing. So you could actually, for the first time, when they did their human genome project, which was at the Wellcome Sanger Institute and the Broad in the States mostly, they were able to work out all the DNA in the organism. And the collaborators at Oxford University that I was working with said that, well, if we can do it in a human, which has quite a big genome, we could do it in bacteria. Maybe we could use that to diagnose diseases. And actually, if you knew the diagnosis, then you might know the treatment as well. If you could see what it was like, if it couldn't be treated by a certain bacteria, then you could have the treatment as well. You could say, well, treat it with this bacteria. And so this is my sequence dress, and I whole genome sequence the Staphylococcus aureus bacteria that lives on me, because we've all got this kind of the normal flora that lives on us, and Staph aureus lives in at least 30% of the population. It lives in the front of your nose mainly. So this artwork is actually the bacteria from the front of my nose. And I whole genome sequence it. I learned the entire process of how to do that, from growing the bacteria right up to doing the bioinformatics to assemble the genome of the organism. And so that's projected behind and onto the dress, which is a 1960 dress design, which is the first time that MRSA actually was found. Is projected the light output of an Illumina whole genome sequencer as it's actually sequencing the bacteria, and so it's flickering on the dress, and the whole genome, which is about 2.8 million bases, is scrolling through behind as a kind of halo around the dress. And this is video mapped onto it, and the dress actually is patterned in the same way as the MRSA quilt as well, but with MRSA, vancomycin resistant Staph aureus, and the Staph aureus from my body, and then also embroidered. And from that, this is the thing I was talking about, the National Collection of Type Cultures. They have this enormous collection of freeze-dried bacteria, and it's freeze-dried for posterity. So one of the amazing things they've got is the world's oldest living cholera, which is from 1915, that came from a soldier who was in hospital in Alexandria in Egypt. And it's actually penicillin resistant. And that's interesting because penicillin wasn't discovered until 1940. Well, it was discovered in 1928, but it wasn't used as a drug until 1941. That's the year it was the start of the antibiotic age. And this is because bacteria are actually naturally resistant to things, and penicillin comes from a mold in the environment. So this bacteria had already developed a resistance to this mold in the environment, which is kind of fascinating. And this one is the bacteria from my body. So it's this one from the front of my nose, the Staph aureus, and I was able to put it into this collection. They have thousands of organisms which researchers can download and not download, but send off or get all the information on, and have sent to their lab to work on. And this one is NCTC 14139 if you want to use it in your future artworks or research as well. And it's the first time they've ever had a bacteria in the collection because of its artistic importance. So I like that, and it's written in all the sort of the annals of the NCTC that this one's there for its artistic importance. So there it is. I don't know if you get QI in America. I think you do on one of the channels there. Is it on the public television? But anyway, it's a very highly syndicated show all around the world. And bizarrely, this made it a question on my MRSA quilt, and there's a picture of me there. Made it onto this prime time BBC show where it was debated. It's kind of clever comedy show kind of thing, where they ask questions and people give silly answers and stuff. But they actually described MRSA being a deadly strain of a drug-resistant bacterium, and how this was a huge existential threat. So this is wonderful for the research because and getting the word out there, because it made it onto this big TV show. I'm also fascinated in things like tuberculosis. This is the world's largest, well, one of the world's largest infectious killers. This year, last year, COVID has kind of beaten it a little bit. But we're kind of used to sort of global about that on the news. So you have to imagine that the death rate annually from TB is about 1.4 million a year. So it's much higher than any other infectious disease. And so it's something really fascinating there. And so I've done a lot of work around this, and this is about the rest cure. This piece is called Rest, because before the antibiotic age, the only treatment for TB was rest. And the TB is curable, it's treatable, and it's almost always curable, but it's not almost always cured or treated, because in huge parts of the world, it's really difficult to get access to treatment and drugs, and there is massive stigma about the disease in some countries, and so people don't even want to be seen to be accessing the treatment and stuff. So it's a kind of fascinating thing. So I made this artwork to tell the story of the rest cure, and it's a tiny hospital bed based on a model that I saw, kind of public health intervention from like the 1940s, where they actually showed people they would bring like school groups and things and show them, this is the bed in the sanatorium, and there'd be the screen around it to stop the breath traveling, and you'd be confined to the bed. And I got really into these old documents and like old public health advice things, which is really, and so I'm also very fascinated in the modern ones and looking back at the similarities as well. They would tell people things like, if you lie down, you take this many breaths a minute, walking about, there's this many breaths, you should take this many bites of food. It was that regulated with it. It's very odd, and a lot of things which maybe your grandparents or great-grandparents would consider to be common sense is actually to do with this like history of TB and this sanatorium movement. And I bet when you're 80 or something, you'll be telling your kids, well, let's open all the windows because, we've got to get the ventilation going or something like that. These things will probably, they will stay with you, and you'll be passing them on to the future. Before tuberculosis, before streptomycin, we had these kind of things as well. A pneumothorax machine, which was used to collapse the lungs of patients, one lung at a time, obviously, to give it a rest from the tuberculosis in order to cure it. And this is an antique pneumothorax machine that I've carved and engraved. So I've carved it with the image of the tissue of a lung that's been damaged by TB with these granulomas and these cavities. And the image on the cylinder that you can see there is what they call the scattered red ribbons of how TB looks under the microscope once you dye it, if you can look at it. It's really hard to diagnose that way. So now they use different methods to diagnose it as well. And they diagnose it with, you can use whole genome sequencing from the sputum as well. And they used another public health intervention was a Blue Henry, this kind of sputum cup that people would use to carry around and then spit into when they coughed up sputum from the TB. And I engraved it with the first genomic proof of a super spreader that was done by Modernising Medical Microbiology. And this was featured in the Lancet journal as well to kind of illustrate this issue. And it mainly affects in like the UK, it mainly affects people with what they call chaotic lifestyles, so homeless people, drug addicts and things like that. In other parts of the world it can affect anyone because they're much more exposed to it. It's got much higher rates. I was in Tashkent a couple of years ago to give a talk for the Médecins Sans Frontières conference on tuberculosis in Eastern Europe and Central Asia, and the rates there are significantly higher than places like the USA or the UK. And in 1902, they used to think that the primary cause of tuberculosis was dust, and that it was the TB was kind of in the dust, or people would spit and it would get collected in dust, and that would be infectious, they believed. It's not. We now know that it has to be kind of coughed and aerosolized. So it's interesting because we've seen in a very short time in the COVID pandemic, this thing of like one thing that you were told to do, and turns out that that's not really a big issue for it. We've seen this kind of, instead of having to wait, like 40 years for this research to come out, now these things are being discovered much faster. So now we know that fomite transmission, like the stuff on your hands, isn't such a big issue as the aerosols for COVID or something. So it was really interesting to see these things, and they had a lot of public health interventions about dust in those days. We've also done work around gut bacteria and human fecal transplants, which is when you take the microbiome, the poop, from a healthy person and put it into a person who's got a Clostridium difficile infection in order to recolonize their gut with healthy bacteria that aren't going to make them ill. So this piece actually contains a human fecal transplant, but it's sterilized because it's in a gallery. It's in the gallery at Eden Project in Cornwall, and you have to sterilize it because otherwise it'll start to ferment and explode. And what you don't want in an art gallery is a human fecal transplant exploding. The images on the box are based on the spores that form in the gut as well, and this bacteria can kind of trap itself in the biofilms in the gut in this spore form, which makes it incredibly hard to treat. And so that's why they came up with this way of treating it, which seems to work, because it's actually a disease that you get as a consequence of taking antibiotic treatments, because the gut bacteria that you have gets depleted, and this Clostridium difficile can grow kind of like a weed all through the gut, and it fills all the gaps, and then it takes over and it causes horrible symptoms like diarrhea and bleeding and ulcers, and you can die from it. So they came up with this treatment, and it's interesting because there's some rules around it. They screen it, but there's some unforeseen effects. So they only use it on people who are really ill. So you can use it. I mean, there's some suggestion that it could affect how you gain weight or not, for instance, depending on what gut bacteria you have. But obviously that can't be used as a weight treatment yet, because we don't know what the effects are. It's not well enough understood. So this piece is called "Don't Try This at Home," because there's actually a movement to do human fecal transplants at home, and there are videos online how to do it, but it's not really a good idea. And another artwork I made was using the gut model at Leeds University, who I collaborated with on the previous project. And this is the biofilms of Clostridium difficile growing on linen lace that we suspended in their gut model, which is this like bacterial ecosystem with different jars and bubbling things and different food going into it, where basically they start it off with a healthy sample of poop, and then after a little while they add some Clostridium difficile bacteria, and then it's all still quite happy, and then they add antibiotics, and then you get this thing called dysbiosis, where the bacterial biofilms on the jars break away, and so it looks like it's all kind of collapsing and there's just holes everywhere where the bacteria should be, and then they regrow, and in that time it's Clostridium difficile. So these are the actual biofilms, and they're grown onto lace, and then this is the artwork that I made from it, which is called Spindle, and Clostridium actually means spindle. There's a whole background to the relationship of lace and Irish linen and linen that I could go into, but it's probably a bit complicated for now. We've only got an hour. I did another project called Make, Do and Mend, which was about, could we heal antibiotic resistance using CRISPR DNA modification? So it's an artwork to talk about these ideas. It's not meant to be a design solution. None of my work's design. None of my work is necessarily providing any kind of solution. It's asking people to think about it, and to join a conversation or to have an artistic experience of thinking about it. So it was inspired by this leaflet by the UK government during the war, which was about unpicking and mending and how to wash things and make decorative patches and patch things. And the image on the other side is Similar to the MRSA quilt, I grew fecal samples of patients in a study in Oxford onto silk, working with Nicola Fawcett here. They're grown on a chromogenic agar, which will grow some bacteria like E. coli will grow pink, and some other bacteria like Enterococcus will grow blue. So a healthy gut microbiome would be a mixture of kind of pink and blue spots. But as you can see, some of the patients are just one species or just another species of bacteria. So some of those people have got like a huge amount of like lack of diversity in their gut, and so the image is kind of we've got the E. coli bacteria there and some antibiotic discs attached to it as well, but it's sort of telling that story. So what I wanted to do was kind of, could I remove an antibiotic resistance gene and cut it out using CRISPR? And I managed to do it. When you edit a bacterial genome, you have to repair it. You literally have to patch it, otherwise, because it's got a circular DNA, and it won't rejoin. It doesn't rejoin like a human DNA or plant DNA will stitch itself back together, but bacteria don't do that. So you have to stitch it back together genomically or genetically. And I did that using the phrase make, do, amend, which was converted from ASCII code into base 4 into the DNA bases and then used to repair the bacterial genome. And they talk about this CRISPR gene editing as being the easy way to edit DNA. And of course it is much easier than it was, but it's still very difficult. And, it takes a very long time and you don't know if your experiment will work. When I did this in the lab, I was working alongside some other researchers and none of their experiments worked. And I was lucky that mine did. And the reason it did was I didn't follow the scientific protocol and pick 20 colonies of bacteria to test. I picked all the colonies of bacteria to test, which was about 70, which was obviously a lot more work. But I didn't want to take the chance of the experiment failing because I didn't have enough budget, because I'm only an artist, to repeat the experiment. So, and one colony of bacteria out of the 70 had taken up the CRISPR edit, and it was the actual finished bacteria that I wanted. And then I used that to patch this suit. This is a World War II women's suit. And during that time, I mentioned earlier that the start of the antibiotic age was 1941. The UK government also brought out at that time something called the utility mark, which was like two little Pac-Man faces, if you know what Pac-Man is. It might be too old for you. Sort of two little C's or pieces of cheese kind of shapes, with a number 41 there. And this was stamped on furniture, clothes, anything which was considered to be sort of sustainably sourced during the rationing period of the war. And so this suit was qualified as this. But the weird thing is that suits and tables and things were considered, these controlled commodities. But of course, antibiotics weren't. And my father, who died earlier this year, told me that he went to the doctors, I think, in the sort of early 50s, late 40s. And the doctor said to him, "Whilst you're here, do you want some of this?" and pointed to a bottle on the shelf. And my dad was like, "I don't know what it is." And he said, "It's penicillin tonic." And my dad said, "Well, what do I want that for?" And he said, "Well, it's very good for aftershave." So can you imagine why we have this issue of antibiotic resistance when they were giving it out as aftershave? And that's not even the biggest problem. The biggest problem is the use of antibiotics in agriculture that leach into the water supply, and they get into all the water, and something like 70% of all antibiotics used in the world are used in agriculture, and quite a lot of the time it's as a growth promoter. So when you see, like, I was quite surprised. I went in a burger restaurant in California, Carl's Jr.'s, and they had one on the menu. They had one burger that was called the All Natural Burger, and it made a point of saying antibiotic free, which meant that every single other burger on the menu had antibiotics in its manufacture, which seemed quite surprising to me. But it's interesting they're stating it, because I bet in other places in the world they don't state it. And I know in Europe they say they don't do this, but I think there's still a thing where if any cattle has the slightest thing wrong with it, they dose them all with the antibiotics that just happen to also be a growth promoter. So it's something, I mean, talking about STEAMivism, I think this is a big issue that we need to work on. This issue of antibiotic resistance is an existential threat, because the antibiotics that we use to treat diseases are stopping working because the bacteria are evolving the ability to outcompete them basically and resist them. So this work, so the suit, I grew the bacteria onto cloth, as I showed before, and I patched the suit using that cloth. So the CRISPR edited bacteria is on the silk that patches the suit from the time, and it's playing with this idea of the controlled commodity. And there are these public health leaflets of the day behind it with these kind of controlled commodity marks and different penicillins and things like that. And it made it onto the front cover of your CDC's Emerging Infectious Diseases journal, which was nice, in 2019. So that was lovely. And I came, I think, just at the end of 2019, that's to New York, to the UN special event, UN General Assembly special event on antibiotic resistance. So I was able to present that work and the MRSA quilt work there. And that was lovely. I've done projects to do with dental bacteria as well, and the history of dentistry and the history of medicine. I won't go into too many details on all this. I've worked, this is me handling Yersinia pestis bacteria in Porton Down in the UK. So Porton Down is the highest security lab in the UK, and Yersinia pestis is the plague. So that's me holding in my hand in a category 3 lab there. I've also had the chance to peer in at their category 4 labs where they study things like Ebola, but I've never worked with them. I don't work with viruses that much. I work a bit with COVID, but I haven't worked that much with viruses. And then through the National Collection of Type Cultures, I was able to learn how to extract the DNA from killed Yersinia pestis, and then I made artworks that are incorporating that. So I've made this piece, which is called Clean Linen, because in the history of linen, they thought that that they would wear linen and they wouldn't wash. This isn't like the sort of back in the time of humoral medicine, before modern medicine, they thought that the linen would leach the toxins out of the body and you could wash the linen. If you washed yourself, you would catch a cold, they thought. So it wasn't good to wash yourself. So this one is embroidered with the images of different diseases, TB, I think we've got scarlet fever, plague, and diphtheria. And then I've impregnated with the extracted DNA of these. And the extracted DNA is okay to work with because it's not the living organism. It's completely safe. And so this piece is my plague dress, which is stuffed full of all the spices and scents and perfumes that they would use, the plague doctors would use, like in their beaks, or they would carry poses to protect themselves. So it's things like turmeric and lavender. And the dress is dyed with walnut, which Culpeper, the herbalist, the famous plague era herbalist, said was a great treatment for plague. The embroidery on it is actual plague era embroidery, so it's from the 1660s, and the dress is a 1665 dress design that I remade. And so that embroidery specifically is impregnated with the extracted DNA of plague. And this is currently on show at the Boerhaave Museum in Leiden, if you happen to be in the Netherlands. I'm watching this, and you can go and visit it. And the Boerhaave used to be a former pest house, a plague hospital. So it's the ideal location for it. And it's in a show called Contagious or Infected, depending on how you translate the Dutch, which was planned years and years before the COVID pandemic. And it was talking about this notion of disease X, which was this coming disease that people said might be happening and that we should prepare for. But obviously not enough preparation took place. The arts knew, that this was going to be an issue, but I don't think the policymakers, which is, I guess, the people you really need to reach with STEAMivism or any kind of activism, is the policymakers who need to have their minds changed. They didn't pay attention. There's another image of it. It was first shown in the Guangzhou Triennial in China, which was formerly known as the city's formerly named Canton, and it was the largest maritime port on the Silk Road. And the original Great Plague travelled from the region like near Tashkent, near Uzbekistan, through China, through the Silk Road to the Netherlands, and then on to the UK. I haven't shown this in the UK yet, so maybe it can make the same journey. And that's it in the Boer Haven at the moment. And that is the King of the Netherlands looking at it. So you can have quite a lot of fun with this kind of stuff. The only thing is the pandemic was on and I couldn't go to the opening, so I never met the King. So it's the second time I've missed out meeting the King of the Netherlands, actually. I talked a lot about the relationship of antibiotics and diagnostics, and if we could diagnose it. And my collaborators at Oxford have now managed to actually prove this. And so what they can do is they can look at the whole genome sequence of somebody's tuberculosis, and they can infer from that, you sequence the DNA, and it will infer which of the first 4 frontline drugs that are used for TB can be used to treat that patient. And this is the first time that's ever been done. So you get the swab of the bacterium or the sputum or whatever, you whole genome sequence it, and the computer can output the treatment, and you know it's the right treatment. So this is incredible research. So not only is this now starting to be used, and it's quite a recent publication, but it's starting to be used. This is a kind of methodology that can be used for lots of different bacteria for the future. So you can maybe foresee a time when we can really target the antibiotic treatment. So we're much more careful about how we use it and really target it. Because probably you've taken antibiotics, but I bet they didn't do all the tests beforehand to whole genome sequence things and prove things. So it's going to be groundbreaking. And we made, myself and Alex May made this installation about it, where the drugs, the different colours, the clouds there are the different drugs. And they had 10,000 people in the study, over 10,000. So each of these little bacillus that you can see flowing about is somebody's tuberculosis in the study. And you can kind of they kind of cluster around you, and you can kind of take them to different bacteria, different antibiotic clouds, and they will disappear. They will get kind of eaten away by them. So it's sort of making clear this link. So there's some other images of it. It's a kind of 3D interactive thing. And another work I made during the pandemic was about the issue of domestic violence during the pandemic. I was commissioned by the University of Brighton to make a piece in collaboration with the charity Frieze, who are a domestic violence charity. And I was interested in the fact that, so there was this term, I think in the States you call it sheltering in place, I think, but over here we called it shielding. So they were shielding the vulnerable. They told like people who were highly susceptible to get COVID-19 to basically not go out, and we'll bring you your shopping, we'll bring you everything, we'll bring you your medicines, whatever you need, but you can't go out. So this was like a terrible thing for some people, but obviously it protected them. So they called it shielding. But with the lockdown, people were locked down maybe at home and they couldn't go out, and they were locked down with partners who were abusive, because there was a huge rise in domestic violence in the early stages of the pandemic. And not just in the UK, but worldwide. I was collecting data from all around the world and articles and things like that from all around the world. And so this piece is about that, and it's based on the images of the temporary hospitals in Wuhan, but they're kind of little homely kind of domestic kind of ones as well, because they've got this embroidery on, and they're using these natural substances to natural dyes and things in it, so it's quite craft-like. But each of the beds is impregnated with the extracted RNA of SARS-CoV-2, which is the virus that causes COVID-19, from a plasmid construct. So it's not the actual, we didn't get the virus and take the RNA out. It's been grown, just the RNA, inside a bacterium. on a plasmid. But yeah, this work is what I made during the pandemic. Some close-ups of it, and so you can see it's quite small. I also made a series of lockdown votives, so there might be some scenes you recognise there. Did you have the massive toilet roll grab in the States, or wherever you're listening? I think there's people from all around the world. We certainly did in the UK. That was probably one of the most famous things, was everybody suddenly went mad and bought toilet rolls. They never foresaw that in the film Contagion, did they? Also the lungs. So there's stories from my friends, stories from my everyday life and things like that of the pandemic. And this piece is about snowflakes, and was kind of playing with the idea of snowflakes traveling and diseases traveling in the air, because there was rumors that SARS-CoV-2 might travel in the clouds and things and then fall in snowflakes or in rain. I mean, I don't think it's an issue, but it was hypothesized and it was quite artistically appealing to me. So these snowflakes were generated with the names of the viruses, the code numbers of the viruses and bacteria in different collections. And there's just to sort of wind up, I'm also working on this project, which is called Fermenting Futures, and working with Boku University. So this is a pre-visualization. We're just about kind of working on the final stages of it to present it at the end of the month. I'm also working with Alex May and the University of Life Sciences in Vienna. I'm working with genetically modifying yeast. And through this project, we've created a yeast which is able to capture carbon from the environment and output lactic acid. Lactic acid can be polymerized to make something called PLA, which is 3D printing filament. So basically it contains and also represents and also has in this, it has the plastic in it, but also is the yeast fermenting in it. So it's called Fermenting Futures. But the idea is, it can eat carbon and output plastic. So that's a piece that we're making now. This is another work in progress, which is called Culture, that's about the idea that yeast has been integral to the development of human culture. And the scientists we've been working with have discovered the mechanisms of fermentation, because some yeast, one yeast evolved this ability to ferment, which meant we proliferated it around the planet. So we've really helped it to grow. We've made it this very successful life form. So we're kind of playing with that in the work as well. And there's an image of the votives that I showed you earlier, and that's in the Valkhof Museum in Nijmegen in the Netherlands. And that's some of my supporters and things like that. Just quickly flash that up. And that's my links. And I would love it if you've got some questions for me just in the last few minutes. We've got a few minutes more, so we can have some questions. So I'll stop sharing. Maybe there's some questions in the chat, or if anyone's got one, just shout one out. Read the questions.
What kind of first inspired you to start doing this? What gave you the idea to combine art and biology in such a beautiful way?
I think it's just because when you're kind of making art, is you're always looking for a kind of subject or something to talk about, and it turned out that just as I started to do that and got more and more into it, just kind of flowed from there, really. I think my earliest works, I was quite interested in HeLa cells in the early stages. And there's this famous story of a woman whose cells were taken without her permission. And then they were kind of proliferated around the world and used in lab research around the world. And that was kind of fascinating to me. But this is we're talking, quite a long time ago. So you didn't have the same kind of access to finding out stuff in the way that you do now. But it was just the more I looked into it and the more I kind of approached people, and they would tell me something and then something else builds on it. So I kind of think about my art practice as a ball of plasticine if you know, it's about the same thing there. I don't know. A ball of plasticine where bits are getting stuck on it, you know, like other bits of coloured plasticine is turning into this big gloopy ball of plasticine that's getting bigger and growing in different directions. And I just kind of follow, like if I'm interested in something, I just kind of follow it and follow it. So I never really consider I've finished a project. So I just am always like looking for the next step in the project. And then so my knowledge of science, because I do all my work hands-on in the lab and I get scientists to teach me. So I'm as much in the lab usually, not during the pandemic, obviously, but as much in the lab as I am in the studio. And I do everything hands-on. And then it's just like the chatting to the scientists and stuff. And it just kind of flows from there. It's just kind of fascinating to me and I'm passionate about it. So I think it's just, yeah, just sort of grew in that direction. I think there's a thing with bacteria where they do something called chemotax. Chemotaxing is how they move. They tumble uncontrollably till they find a better situation and then they stay there for a bit. And then when the situation, you know, the food's run out or something, then they tumble on. And I think art is a bit like that. Art practice, freelance art practice is a bit like that anyway. I don't know if that answers your question. I hope it does. So I was looking through if there's any other questions. So any other questions, just shout them out.
I think we have a couple of questions and then I have a few questions for you. Yeah. So we had Elizabeth, who is one of our climate activists who's speaking today, actually. And she is asking what your advice is to a young person who wants to use their artwork to join global conversations about improving the world.
I mean, social media is very good for this if it's global conversations, and sharing it on things like Twitter with all the hashtags or Instagram. I think Twitter's really good because then you can reach out to scientists. I think a lot of scientists are on Twitter. And also, don't be too snobby about LinkedIn. I've had some fantastic opportunities and conversations and meetings with people and stuff through LinkedIn, which and sometimes the people in the art world laugh at me that I'm on LinkedIn, but then, you know, the joke's on them because I got a show in Texas from that and I'm only in England. They funded everything and it was all wonderful. So, you know, so I think that's important. I think it's getting it in front of policymakers as well. So I do a lot of projects that involve the European Union and things like that. So whenever I think there are going to be policymakers or people that can have an impact, and get them thinking about it, because they usually only have time to read the top paragraph of a report. They don't read the whole report. So if you hide anything intricate in the report, they don't see it. If they see an artwork that kind of entices them in, then you can start a conversation. So starting conversations, and if you don't have access to be able to go places in the early stages, but you might later, you know, it's good to start those conversations online and kind of engage that way.
Thank you. Hopefully that answered your question, Elizabeth. I had a couple of questions for you. I'm very interested in genetics as well, and I've done some work sequencing genomes. So I think it's just amazing that you're taking that and really making an impact through making artwork with genetics and bacteria. So, you know, as we can see, technology is advancing at just an exponential rate. So, you know, CRISPR, gene editing, all of these things, I think there's so many bioethical implications to, you know, everything that you're working with. So I would say, you know, what are some of your fears regarding genetic editing in the future?
Well, one of my fears is that people will still resist it so much that the benefits can't be used, which might sound a bit strange because that's a big concern, especially in Europe. Because with climate change, things like the gene editing of plants, the sort of cisgenic modification of plants where you use knockouts rather than actually transgenically modifying them, it's something that could happen in nature, but it happens much faster and in a much more targeted way. And then you'd be able to kind of make plants that may be more drought resistant or something like that, which is actually beneficial to preventing like the impacts or mitigating some of the impacts of climate change. So one of the things I'm actually interested in is getting people to think about this issue of natural or unnatural, because there's this phrase natural that's bandied about whether something's natural or not. And it's very interesting if you start looking at the definition of this, like, is cabbage natural or sprouts natural? They all come from wild mustard. Wild mustard's the natural thing. Everything else has been so altered by crossbreeding and growing that we have like about 10 different vegetables, cauliflower as well. It's the same, they all come from this wild mustard. And so they're so different. It's like, what is this thing called natural anyway? And, you know, like in the EU, you're not allowed to eat genetically modified food. So humans can't eat genetically modified food, but that law doesn't apply to animals. So you can eat animals that have eaten genetically modified food. Is that natural? You know, so I have these concerns about it as well. I think it's something you don't want to put the genie back in the bottle because we're worried about a grey goo situation. Like, you know, they talk about nanotechnology gone mad and everything. The nanobots turn everything into grey goo, but it couldn't really happen like that. But that's the sort of the fear that people talk about. But it's the same thing with antibiotics. Yeah, we have this problem with antibiotic resistance now because they were misused, but nobody really knew what use was. Alexander Fleming talked about the idea that bacteria could develop resistance and that you should finish the course, but he never said what the course was, and the course was never tested on women. The course is not tested on children. It's tested on men of a certain body build. So now we also have things like personalized medicine and stuff like that. So I think things get used in all sorts of ways. And so what you've got to do is involve as many people in the process of thinking about what we should do, so we kind of mitigate for that. But I don't think we can sort of say, no, we don't want to do this, no, we don't want to do that, because it is so useful and it has so many potential benefits as well.
Definitely. I think everything you talked about is so also applicable to you know, what we eat in today's society. COVID-19 as well, I think you touched on that in your presentation. And I mean, I just think it's amazing that you're using your art as a vessel to advocate for a better world and just participate in activism. I think it's just such a wonderful avenue to do so. We'll take maybe 2 more questions. Shreya, do you have a question? I think you had some questions.
Yeah, I was also kind of wondering how like you decided to go the formal scientific route when approaching your art instead of maybe doing more of like a self-study. Like, I know that you worked in labs to develop your various art pieces. So like, why did you decide to like specifically go like, I guess, kind of like academia and like the science route instead of like self-study, or maybe like representing the different scientific aspects in a more abstract way instead of like the concrete, like different genomes and stuff like that?
I mean, I do like the DIY bio stuff as well. So you kind of do what you do. It's like any means necessary. So to make work, I'll do DIY stuff. I do DIY workshops as well, like how to grow bacteria safely and culture bacteria at home or something. Like I've done workshops with GenSpace in New York and places like that. So so I definitely do that. But I'm really interested in these like very cutting edge sciences, and they use expensive tools, and it's very complicated science. So the stuff that I'm interested in working on isn't available in like the DIY bio kind of labs. It's just they don't have it, and they don't have the skills really. I mean, some people do, and there are some amazing people that work across academia and DIY bio, or you know, biohacking or whatever. So there are people working in that field. But also it's just, for me, I just wanted, actually it's a kind of like, I go in and I kind of see how the scientists work and what scientists are like, you know, like how close is the representation in like the media or something to what a scientist is or what a scientist does. And so I'm interested in that too, rather than just doing it myself. And I guess with the abstract stuff, like, I mean, I guess I did paintings about, like, I used to do paintings based on microscopy and things like that. This almost sort of predates the sort of the access to the web stuff, because I would get books with microscope images in and paint pictures based on them and stuff, because I trained as a painter originally. So I've done that, and then I guess I moved on, because it's all a process. You're looking for the next interesting thing and the next step, and then just more and more exploration, I think.